Cette
what's going on
conférence va
with
maintenant être enregistrée.
holobiont. So sorry. I think welcome everyone. I'm hearing people coming in, and now I'm officially there. So, as I was saying, I'm researching the holobiont, all these, for instance, these algal organisms, and interest in their resilience to global change. And I'm using the omics as a tool to investigate this change, investigate the metabolic change during, for instance, perturbation or change of the system. And hopefully study wherever the change, the system, these holobionts can go back to the initial phase. Okay, and more with more focus now on the algal holobiont, I work on phytoplankton, which are an essential component of aquatic ecosystem. You can see here on this image algal bloom. Seen from space, these are millions of cells that divide and form this gigantic population. And they contribute to the carbon fixation, so they have a very important role for the ocean functioning, but also for the biochemistry, the cycle of elements on Earth. And what these algae, of course, do not live alone. They will bloom and live in interaction with other microbes. And we can look at the population as multiple cells. And of course, these cells will not be homogeneous. There will be some heterogeneity in these blooms. And for instance, some of the algae can become infected and might be actually intruder cells. We have the example of algal models we work with in the lab, and pathogens such as other opportunistic microbes can infect these cells. For instance, bacteria, virus, or other single-cell eukaryotes that we will put them in the group of protists. But nonetheless, this protist, of course, it's not a taxonomic clade. You will see now some example I work with. And I want to emphasize that these parasites really contribute to the carbon transfer in these food webs because they will interact with the cells. They will interact also with bacteria. You have here an example of fungal shunt, a new concept that has been published last year, showing that that the parasite really contribute to bring back some carbons into the food webs, not only by infecting, but because infected cells can be eaten by the grazers, and then, of course, contribute to this food web. And if we look more closely at now the plankton model I work with in the lab, I'm interested, for instance, in toxic algae such as Alexandrium minutum that can form these huge blooms. This looks kind of cool on the first point of view, but considering that some of these cells produce toxins, especially, for instance, the group of saxitoxins that are neurotoxic for mammals, this is actually a problem when you have this massive bloom. And if we look more closely, sampling at this bloom with a microscope, we look at the cells. We have here healthy Alexandrium swimming, but also some infected cells. So I'm working with a parasite from the genus Parvilucifera, that is a generalist parasite that can infect many clade of dinoflagellates. Interestingly, these parasites actually are very close taxonomically to their host. That's due to the coevolution between host and parasite in ocean. And what you see here is the last stage of infection when you have these little new spores that are released in the environment and that will try to infect the swimming cells, swimming healthy cells. So these parasites are very interesting because they also can infect toxic algae. So one of my research questions is to understand whether saxitoxins are involved in the infection process. I'm also working now with another model of parasite infecting this time the diatoms. The diatoms groups in another big group of phytoplankton, in particular Cocinodiscus granii. For instance, you have here an example of healthy cells And I have isolate and work now in the lab with no mycete parasite from the name Laganisma coccinodici, but can again infect the cell and until the last stage, the reproduction of this parasite. And you're seeing here again the release of all the zoospores that are now swimming and has to find quickly a host because this life stage is short-lived. So you've just seen two models of organisms I'm working with in the group of dinoflagellate and diatom. So you've just seen the release of these zoospores. So this is the free flagellated form of the parasite that will infect the host. And then it's much more complex because you can have different, let's say, different stage of infection. For instance, the feeding stage, where the cell and the parasite start to get to develop within the cells and eat alive, let's say, these cells. And why? Because then they want to reproduce to reach what we call the sporont, the moment where new spores will be produced within the host. And what is very interesting is that chemicals, we have not so many studies, but some exist showing that chemicals are involved in this infection process. What is also interesting to note is that these parasites are parasitoids, so they always kill their host. They need their host for surviving, so that's a bit tricky to handle in the lab. And I'm basically interested in my research about mechanisms of infection, of how the host is infected, how the parasite defends itself, what chemicals are involved in the attraction from the parasite to the host, and last but not least, big topic about how hosts can defend against these parasites. And for all of these different research questions, our postulate is that chemical cues are involved. And this is to show you that we don't know much, but they basically always appear at the last stage of the bloom, these parasites. And we don't know much about these mechanisms that are involved. And we start working, when I start my postdoc, I start developing and working with the model of the mycete infecting the diatom using the co-cultivation chambers, where basically you can put different population exposed to each other. For instance, in my control culture, healthy population, and in my treatment, I will put infected population versus exposed cells. Exposed because they are healthy, but they are exposed to the exudate released by the infected population. Because this co-culture chamber, you might know for most of you that they are separated by this membrane where exudate can pass through. And using high resolution mass spectrometry and equally a comparative metabolomic approach, what we were doing was to sample over time this population and investigate the metabolome change during infection, exposure, or this healthy. And quite interestingly, seeing for instance, using this principal component analysis, shown that there is a clear discrimination between these different populations. And further, by elucidating and looking at the compounds that are associated with infected cells, we identify a couple of chemical class using spectral similarity matching with standards, but also with public database. That's how we came up to identify alkaloids from the beta-carbolin family that were associated with the infected cells. And then by doing the quantifications based on LCMS and further two other analytical methods to confirm it, we could show that these compounds are actually produced by the algae. But during the infection, they will be overexpressed, they will be overproduced during this process. So then to prove the function of these compounds, because we had access to the standard, we then proceed to do some functional assay by adding the compounds to the healthy culture and showing that at the relevant concentration, the growth rate was reduced. So the healthy cells do not grow much when you add these compounds. And on the other hand, when you put this compound in infected culture, we see the infection progressing much faster, especially with one of them. And this led us to conclude, to show that these compounds are involved in the infection. They will inhibit the growth, and of course, because the cell population is smaller, this population will be infected faster. So of course, we then wanted to know whether these compounds were just a one-time occurrence, or if we could identify them again in a bloom community. So in just before Corona, we had the chance to again sample from the same site in Germany, Helgoland, that is a German island, and we found we had to isolate again Lagenisma and a couple of other co-occurring algae, notably Cocinodiscus. And then using again the comparative metabolomic approach, we could find that most Cocinodiscus species were susceptible, they could be infected, and their cellular metabolome was tremendously different when they were infected. And within the population, there were also cells that were also strains that were not infected, but were what we can say resistant. Interestingly, the cellular metabolome were not so much changed, excluding for one of the Cocinodiscus species, Cocinodiscus wellesii, and we postulate that maybe the exposure to the parasite triggers some compounds related to the resistance. So further, we were looking, of course, at our target compounds, these alkaloids, and we found them again, especially in this highly susceptible Cocinodiscus granii. This led us to conclude that the beta-carbolins, these compounds are actually hijacked by the parasite. The parasite will use the compounds of the host to its own purpose, and it's a conserved mechanism because we find it again in a new bloom. Okay, and we are now, of course, mining this data set, notably using molecular networking to identify other compounds of interest. We are, for instance, targeting a signaling molecule that is known as a plant apocarotenoid. So now we are thinking that this compound is involved in the host defense. You have here identification based with the MSMS, a comparison of the MSMS experiments with the standards. And of course, this basically is it. What we have so far. We are still looking at the functions, so this will be for the next talk. But I'd like to present you today also one of other methods we have developed to address cellular heterogeneity. As I said before, in blooms, you can have huge heterogeneity. For instance, you have here example of an infected cells, and usually all cultures are not synchronized. So you would see cells in various stages, from completely empty frustule to healthy to infected with a different stage together. And of course, when we do simple batch bulk metabolomics, we will just get an average of this population. So to prevent that, we developed a method using a single cell metabolomics to discriminate cells with a particular phenotype. And of course, I use the cells that were infected with these two parasites and so on and so forth. So our workflow was as such. The study design was to have cells that we could isolate, and identify whether they're infected and not infected. They were prepared by simply putting them on GFC weight filter, and then we acquire single cell metabolome using MALDI Orbitrap, and using then a statistical analysis based on the classification, we then were looking for biomarker. And with that, we used our first method using the method, as I said, but we put our cells on a GFC filter, so the cells are still alive and immediately analyzed by cell disruption with our laser. And this is done at atmospheric pressure, so there's no disruption of the sample over time. And you have here every single cell that we can look at because there's a built-in camera on the mass spectrometer. Then, of course, as I said, we're using multivariate statistics to search for this biomarker. And with that, we found and identified compounds that are associated with the infection of the diatom and the dinoflagellate. So, for instance, we found theophorbiate as kind of, I think, a degradation pigments that usually arise from chlorophyll. And in the case of a parasitic infection of dinoflagellate, we also found a couple of compounds related to the purine salvage pathway. And this was done in complement by analyzing standards and, of course, confirming the identity of this accurate mass that we are discriminating our dataset. Okay, I hope, I don't know what time is it and where am I, but I hope I did not spend too long over the time, so you can listen to the exciting next talks. But that's it for my part. I'm studying this bloom forming algae. We have now in hands our model organisms that we maintain and we can play with. We have now resistant diatoms that we also are studying. We are also interested with the parasites that infect toxic dinoflagellate, and all of this, yes, we hope with our tools that we can identify more compounds that are involved in this interaction. And we have now this different approach that we can use for diagnostic. And the idea will be that we can go back to the ocean and be able, of course, to identify and follow these parasite epidemics directly in situ. Okay, so I want to thank you very much. I hope you have some questions, there's some times. And I want to take this opportunity to say that we still have one postdoc position in our group, a contract that goes from now until 2025. So if you know anyone who will be interested in joining our group, please send them to send us my email or Georg Sponert email, please. Okay, so thank you. I want to see you, so I will just try to stop this. Where are you? So I don't see anybody. Okay.
Thanks, Marine, for your talk. I'm always very impressed with all the single cell techniques. So if we have some questions, you can write them in the chat and we will ask them to Marine. Okay, maybe so. Just one question: if so, with this single cell technique, is it easy to have some clue about the concentration of your metabolites in the cell, or?
So we did that for the beta carbolines. We could do a relative quantification. That's the plot you could see over here. So you cannot get the absolute quantification. So that is not possible. But because we had the standard, if you have the standards, you can approximatively say, okay, we have this relative quantification. So we have done quantification based on single cell with our multi-orbit trap, and that's the result we have here. But I will say I will always, if possible, use another complementary methods like basic LC or GCMS, depending the compound you're looking for, because to confirm it, because this will never be absolute quantification. What was great with this class of compound is that they are also can fluoresce, so we could use that property and did the quantification also using a confocal laser scanning microscopy. Yeah.
Okay.
And I would say it's also to finish on this question. I would say it also depends because with you have to understand, and I guess you know that with a MALDI you cannot necessarily ionize every compounds. So there's for sure a lot of compounds we are not detecting with this method. So there's some positive sides and some caveats with the technique.
Okay, thanks. If I can, one more question.
Yes,
can you
of
use
course,
it?
because I think I see a lot of people, but I think maybe a lot of you can send me a question by mail later if you have new idea tomorrow after thinking overnight.
Okay. we have one question in the chat, so maybe I'll ask mine after. So the
Yes.
question is, do you have transcriptomic correlation with your metabolomic data?
Yes, so this is something oncoming with my new grant. This is part of a research now that we don't only do metabolomics but transcriptomics with our analysis, and we already start doing some experiments regarding that, because this will be indeed fantastic to confirm what we're seeing.
Okay, thanks. Maybe we're gonna go with this. I mean, the metabolic signature was already proposed by transcriptomic. It's a question.
Not yet. So we have actually our collaborator. We have a professor in Senckenberg University. Okay, I understand now. I think it's different than what I want to say. Yes. So now we know actually that there is this class of compounds in this model. So I'm actually in touch with Genoscope. I have started a collaboration there where we hope to look for indeed the gene and do transcriptomic of the analysis. I think what is missing is that with a single cell, you cannot get, you know, what you can do when you have LCMS and you do untargeted MSMS experiments with this DDMS, for instance, with this data-dependent mass spectrometry. You cannot do it on one single cell. This means that we are not yet at the stage where we can just plug in MSMS from single cell into GNPS, for instance, and look for a novel compound or compound and target that. So I will say that, so for now, I think transcriptomic will give a more, it's more costly, but it will give a more let's say, group approach to find new gene. I hope this answers the question.
Thanks, Marine. We can go with our second speaker, Eva.
Yes. So I would like to.
You can just close your screen and issue the.
Yes, I just. I'm so sorry. Attends. Stop sharing. Voilà. Okay. Thank you all.
Samuel, would you like?
Let's introduce the next speaker, who is Eva Ternon from Villefranche, in the south of France. So she will speak about chemical interaction and proliferation of toxic harmful microalgae. I don't know if you are here. Yes. So the floor is yours, Eva, for approximately 15 minutes. And thanks a lot for accepting to speak about this field today.
We cannot hear you, Eva.
Is it better now?
Yes, yes, perfect.
Okay, great. Well, thank you very much for the invitation. And I'm going to carry on with microalgae, with a specific group called harmful microalgae. And all the results I will be presenting today are resulting from a collaborative effort during a French national project called Ocean 15, and then a European project called Micro. So as Marin already nicely introduced, microalgae are microorganisms that play a key role in the ocean, in the functioning of the ecosystem. But in some cases, when some conditions are in place, they can form really dense proliferation. And if you take like a drop of seawater at that moment, you will see that most of the biodiversity is represented by one single species. And because they have a very high pigment content, the seawater becomes colored, which leaves like nice colorful waters. But at the same time, these really dense proliferation are also a threat for the ecosystem, resulting in massive die-off of vertebrates and invertebrates. You can see on the picture, there is a massive die-off of fish that were washed up on the shore. From a human perspective, these events can also be a huge loss of money. You have like an example of bloom of Alexandrium that occurred in the eastern USA, after which the loss of up to 18 million in gross revenues in fisheries was calculated. So why is this massive die-off occurring? Because the high photosynthetic activity of these microalgal cells and the high respiration during the night can often yield to a combination of hyperoxia, and then anoxia in the seawater, and drastic decreases in the pH, which is really harmful for most of the organisms that are present in seawater. In the case of proliferation of dinoflagellates, toxins are also involved in this die-off. You can see on this figure that dinoflagellates, the group in pink, is the one that produced the most gene cluster for polyketide biosynthesis. And these polyketides, most of the cases, are toxins. So how harmful algae shape their ecological success? Like any other algae, they're interacting with diverse organisms from the ecosystem. You have like bacterial interactions, interactions with predators, and interactions with other microalgae that occupy the same niche in the ecosystem. In the case of the specific species Ostropsis ovata, which is a benthic microalgae, meaning that it grows on a substrate, you also have the interaction with the substrates, which is often microalgal species. So because we only have 15 minutes, I'm going to focus on the substrate interactions and the competitive interaction in this talk. So we're going to start with substrate interactions. There were several observations in the field showing that Ostropsis ovata preferred substrates, brown algae as a substrate, like Alopteris scoparia or Dictyota species. But on the other side, another study showed that when testing allelochemicals interactions between algae and Ostropsis, the Dictyota dichotoma, the brown algae, was inducing growth inhibition, a decrease in cell abundance, whereas other algae like red and green algae were not producing that effect. So we were wondering at the first place whether the chemistry of brown algae could control the growth or not of Ostropsis ovata. So to test this hypothesis, we used 5 different brown algae species from the Mediterranean Sea. We went to the field and measured the cell abundance of different microalgae, including Ostropsis ovata, on the different species. And we saw that on Dictyota dichotoma, the Dictyota dichotoma had the highest abundance of Ostropsis cells. So that was in line with previous observations made in the field in the same area. Then we came back to the field and sampled algal tally for the different species, and we dipped very quickly methanol, the tally of these different species. The extract in methanol was used for 2 different things. One, first to assess the effect of the surface extracts on the growth and the physiology of the microalgae Ostropsis, and then to perform untargeted metabolomics. So here you look at the results of the bioassay. So on the upper panel, you have the birth rates of Ostropsis, and on the lower panel, the photosynthetic efficiency of the cells. And what we saw is that the highest effect on the microalgal cells were obtained with Taonia atomaria and then Dictyota dichotoma, which is different from which is counterintuitive, given that highest abundance were observed on Dictyota dichotoma. Then if you look at the comparative metabolomics analysis, you saw a clear discrepancy between the two Dictyota species here that are related, and then the two other species, Padenia and Taonia. So that was interesting to see that they have different chemical diversity. Chemical features that were showing up for the Dictyota species were diterpene derivatives, and that were identified thanks to in the library in the lab. And the chemical feature obtained for the Taonia species was a glycerol compound, also obtained through library comparison. And for Taonia and Padenia, the dimethyl sulfate propionate, DMSP, was observed as one of the main chemical features as well. Since we are seeking a compound that would explain why the bioactivity of the Dictyota species, of the Dictyota dichotoma, that is not present, that should not be present in the Dictyota spiralis species, the diterpene could not be our candidates. Same, DMSP is highly produced by Ostropsis ovata itself and cannot be an inhibiting compound in this case. So the glycerol compound could be a good candidate, but we were not able to test pure compound at that time, so we don't have any confirmation on that. We further performed a molecular network on this data set, and we identify a cluster here, cluster J, where one compound could be identified as a candidate for the bioactivity observed for Dictyota dichotoma, which was not shown to happen in Dictyota spiralis. So at this stage of the study, we can tell that, yes, surface extract from microalgae can have an influence on the settlement of Ostropsis and its growth, but it's probably not the main factor of control, and other biological control probably happen, like predation or even the valley shape, or competition with other microalgae as well. So competition with other microalgae is competitive interactions. So again, we started from a field observation where during summer we had a high proliferation of Ostropsis, during which we had like a stable abundance of other microalgae, and the other microalgae started to bloom after the decrease of Ostropsis abundance. And this was had been already observed by other authors, that the Ostropsis bloom was affecting the community of microalgae. So our main question was, is Ostropsis ovata able to control the growth of other microalgae? To test this question, we performed a co-culture experiment using co-culture chambers as marine. The sun is low. Sorry. As Marine presented earlier, so it allows the diffusion of cells and for cell growth and photosynthesis again, and then at the very end of the experiment, we extract the medium and the cells, perform metabolomics analysis by mass spectrometry. So we were very surprised to observe that over the course of experiments, the abundance here of cells diatom didn't change at all between the control and when it was exposed to Ostropsis. There is absolutely no growth inhibition on the diatom on the computer, and there is no contact between the cells. On the other hand, for experiment, we saw a clear decrease of a significant decrease of the abundance of these cells, but they were exposed to Lichmorphora paradoxa, the diadem. I'm sorry, I'm trying to be closer to my computer, and you can hear me well. So that indicated that the diadem clearly inhibited the growth of the dinoflagellates, and the stress was even more observed on the production of toxin by the dinoflagellates. Here in gray, in light gray, was the production of toxin when the cells were exposed to the diadem, and there was a significant increase in toxin production. When we looked at the exometabolome, the chemical diversity present in the cell in the medium after the experiment, we were able to identify a few biomarkers. Fortunately, at that stage of the experiment, we were not able to identify structures, but we put aside that they could be either fatty acid or oxylipins, because those scopins are known to be produced by diatoms in high content, and be bioactive against a large variety of marine organisms, and in particular Ostropsis, as it was shown by Gary et al. In 2016 and 17. During my recent project, we tried to isolate one of these bioactive compound, and we indeed found that there was a small fatty that impair the integrity of host groups to find the final structure of the bioactive compound by purification. So in conclusion, the interaction between Ostropsis and the other members of the ecosystem are still need to be really assessed. As part of the substrate interaction, we know it can exist. There is a potential for a chemical inhibition or chemical attraction, but we don't know its importance, and we don't know the chemicals that are involved in this interaction. For the competition aspects, we did find that other group of microalgae can inhibit the growth of Ostropsis and increase in content by inducing a stress through the production of small fatty acid. But there are other macroalgae that are in play here in the ecosystem, and we were able to show that it's not always the case with all macroalgae. It's a specific interaction. During the chemical project, we also looked at the interactions with bacteria, and this is still analysis that are still in process. And predation was also looked at by a number of groups, but with more an ecological. And with that, I would like to thank a lot all the collaborators that were involved in these different studies, and thank you all for your.
Thank you, Eva, for this nice presentation. Does anybody has any questions? You can write it down on the chat, or either maybe speak it just by turning on your camera. I have one question about, you know, the last the fatty acid that you identified. It's not a compound which is highly water soluble. So how do you explain that this compound has such an importance in the interaction?
It's not highly water soluble, but we were able to purify it from mixture of water and acetonitrile, so it's not that insoluble either.
Okay, so it's still sufficiently soluble to.
Probably, that's what I think. Yeah, but so far we were not able to find it properly in the medium. I think it's related with the last VIP that is in the table, because there is only like a H2O difference. But we're not we didn't really see it in the medium yet.
Okay, thank you. Marine has a question for you.
Yes, Eva, thanks a lot. Because actually that's the first time I see part of your research as a talk, so it's cool. I like it. And so during your co-culture experiment, I suppose nothing was arsenic, right?
No.
So did you see like bacteria appearing, like benefiting also from this co-culture? That's one question. And the second question is that because I saw it's part of the next objective of the project to look at the role of these bacteria, do you expect that they have a sort of contribution to the competitive effect you have seen?
So for the experiment performed, we didn't really see any difference in bacteria communities across experiments. So they were quite stable, and I mean there were no difference between like treatment and the control. So, but on the evidence analysis, we didn't do any genetic analysis on this experiment. As for the other part question, the bacterial interactions. So what we tested there was more about within the competitive interaction. It was more the impact of bacteria on development of ostropsis itself. So with the arsenic strains of ostropsis and bacteria on the other side, to see whether bacteria were kind of compound bacteria were just providing to ostropsis to make them grow and proliferate.
Yeah, okay. I'm looking forward to see more from that part. Thanks a lot, Eva.
Thanks a lot. Does anyone has any more questions? I think maybe we can. Yes. So Christelle asks the question that if the bioactive compounds that is related to the settlement of the microalgae is an abundant major compound or a very minor one.
Sorry, I was away. You mean for in the cells or in the medium?
Let's say both.
Let's say okay. For the medium, I don't have any answer. For the cells, yes, it's one of the major compounds. Otherwise, I mean the purification, yeah, it was easily purified and with a small quantity of cells. So we were lucky, I would say, but it's not always the case.
Okay. Thank you, Eva, and maybe Nathan, you can introduce the next speaker of the afternoon.
Yes, do you hear me, Samuel?
Perfect.
Yes, okay, so for the next presentation, we welcome Benoit Paix. First of all, thank you for presenting today at this RFMF webinar. It's easy for me to introduce you because we were in the same laboratory during 2 years. So you did your PhD at the MAPIEM laboratory at the University of Toulon. And now you are doing a postdoctorate in the Naturalis Biodiversity Center in Leiden, Nederland. So the place is yours. You can start whenever you want.
All right, thanks, Nathan. Yeah, now I can share my screen. And yeah, it's not easy with All right. So thanks everyone. Thanks for the invitation for this webinar. I'm really happy to be able to talk about some results from my PhD about the effect of copper and temperature in the interaction between a seaweed, Taonia atomaria, and a surface microbiome at the surface of this seaweed. So the algal surface is the zone of interaction between the surface of the algae and the microbiomes. And in this microbiome, we can find, like Eva presented, a lot of microalgae like dinoflagellate or diatoms, but also a really high diversity of bacteria. And this algae and its microbiome are forming what we call an holobiont system. And the microbiome can have a really important role in the adaptation and fitness of the seaweed. For example, for the green algae Ulva mutabilis, some bacteria can be chemoattracted and can induce the growth of this seaweed. The environment also can play a really important effect in these interactions, in these chemical interactions between the algae and its surface microbiome. For example, the increase of temperature can decrease the chemical defense of the algae, but also increase the density of the pathogens at the surface, and resulting in a bleaching of the right algae. Other parameters like irradiance, salinity, and pH are also quite important. And for example, you can have an effect for the brown algae Fucus vesiculosus and its microbiome. But when I started my PhD, little was known for Mediterranean models. And that's why I started working on the brown algae Taonia atomaria, which is widely distributed in the Atlantic and Mediterranean coasts, and also really easy for the identification for within the Dictyostacea family. Some of its surface metabolites has been already studied before my PhD, especially with some compounds like the fucoxanthin, but also some sesquiterpenes, and also this compound that Eva told about, the geranyl glycerol. Some of these compounds which were isolated were also tested against some bacteria, and some of them have a really interesting effect, a selective effect against some specific bacterial strains. So I started my PhD with this hypothesis that there was a chemical control of the epibacterial community at the surface of this algae through the production of some of these surface metabolites. So my objective were to understand what is this microbial specificity at the surface of this algae, and what are the roles in this specificity of some specific surface metabolites. And the second objective was to understand if some environmental parameter could influence these interactions, these host microbiota interactions. And here for this talk, I'm going to present two parameters, the temperature and the coppers, which I studied through spatiotemporal monitoring in the Toulon area. So we collected the algae during the whole occurrence period from February to July. And we collected this algae at different sampling sites. The first sampling site is within the Toulon Bay, which is highly anthropized with a lot of contaminations in terms of metals. And the further sites are less anthropized, much more preserved, especially within the Porquerolles National Park, especially for the Porquerolles Island. So we we collected all the different sampling types, seawater, the surface, the bacteria at the surface of the rocky substrates, and of course the microalgae. From the water, we measured all the environmental parameters, and we performed a metabarcoding approach to study all the microbial diversity, so all the bacterial diversity in the 3 types of samples. And parallelly, we also performed a metabolomics approach to work on the surface metabolome of Taonia atomaria using LCMS experiments. And the idea at the end was to combine all these different analyses using a multi-omics analysis and to correlate all the different types of variables. So as the first results, we can observe actually that there is a really high level of copper contaminations in the site S1 located inside of the Toulon compared to the 4 other sites. For the bacterial diversity in our samples, what we observe with this multivariate analysis is that we have clear difference of diversity within the 3 different sampling types: the seawater, the surface of the rocks, and the surface of the algae. And when we focus at the surface of the algae, we can see that there is also some interesting patterns about the diversity. There is first temporal differences, which are explained from February to July, which are explained by the temperature increase. But there is also spatial differences, especially with the site S1, which is highly contaminated by the copper, and also in a lesser extent the lead. If we look for the discriminant taxa, which are discriminant according to the different sampling months, we can find first the taxa which are really specific for the first life stage of the algae in February, which is a really pioneer microbiota. And this microbiota is actually known to be core members and highly specific to the host. And when we go further over time, we can find some taxa which are more expressed in July, with, for example, the Rhodobacteriaceae, which are mainly affiliated to the marine Roseobacter clade, which is really interesting because they are known to use the dimethyl sulfopropionate, the DMSP, as a source of carbon and sulfur. When we look at the taxa which are really discriminant according to the different sites, here we can find some interesting taxa which are really specific to the site which is highly contaminated in copper. And these 2 taxa are interesting because they have already been observed in the surface of algae in highly enriched copper environments in Chile. And these taxa are quite interesting also because they are known to produce some antioxidants, which might be interesting against this type of contaminations. So now when we go into the details of the metabolomics analysis for the surface metabolomes, here with this multivariate analysis, we can see that there is also a temporal shift of the surface metabolomes. And when we look into the biomarkers associated to this temporal shift, we find mainly the DGTA, which are membrane lipids. And these membrane lipids are known to be more produced during the growth of the algae. We find also the DMSP, which is this algal osmolyte, really important in the communication between the seaweeds and also the algae with their prokaryotes. When we do this time a discriminant analysis to look into the difference between the different sampling sites, Here we can find that the site S1 highly contaminated in copper is quite different from the others, and there is some biomarkers which are overexpressed in this site compared to the others. These biomarkers are actually beta-carotene, fucoxanthin, and derivative of the pheophytin, so 3 photopigments. And this is really interesting because the copper is known to have to induce photoinhibitions on the seaweed and also an oxidative stress. And the fucoxanthin and beta-carotene are 2 compounds which can act as an antioxidant against this copper stress. When we do multi-omics correlation networks, so we obtain this type of results. So how this network was constructed, was built with the metabarcoding, the metabolomics, and the environmental parameters datasets. And the idea is to combine all these datasets together and to look into the correlations, the most discriminant correlations between the variables according to the sites and to the months. But of course, we have to be careful when we analyze this type of networks, because we see a lot of links, and all these links, of course, are not necessarily causality links. So when we go into the first cluster on the left, we find first the temperature. Associated to this temperature, we can find also all the membrane lipids, the DGTA, and we can find the Roseobacter taxa, which are known to use the DMSP as a source of carbon and sulfur. And this DMSP, we can also find it in the cluster. On the second cluster on the right, we can see this time the copper. And the copper, we can see some associations with our photopigments, like the fucoxanthin, the beta-carotene, and a derivative of the fucoxanthin. And we find also this taxa, the Rosebacillus, which is also known to produce antioxidant against this type of stress. So there is some conclusions that we can make about the temporal effect first. So when we are in February, we have a young algae which has a pioneer microbiota, and when we go over time to July, we have, of course, the algal growth, and this algal growth can be seen with another expression of some membrane lipids like the DGTAs. We also have the increase of temperature and irradiance, and this is known to induce another expression of DMSP at the surface of the algae. And this DMSP actually can be used as a source of carbon and sulfur by some new colonizers, the Roseobacter clades, which will colonize the surface of the algae. So the effect of the copper here on the left, when we are in a situation where we have a low level of copper contaminations, we have basically normal situations. But when we are on the right, with high copper concentrations, these copper concentrations can induce an oxidative stress and photoinhibitions on the algae. And this stress, the algae will fight against it by overproducing some pigments, photopigments, but also antioxidants like the fucoxanthin. And this fucoxanthin is really interesting because we know that these compounds at the surface can also have an anti-adhesion activity, like some bacterial communities at the surface of the algae. So there is maybe an indirect effect on the microbiome, but also we have to consider a direct effect of the copper on the microbiome with some taxa which could be favored due to their own antioxidant strategies. So as a conclusions, the multi-omics was very interesting because it can help to better understand the complexity of the holobiont system. We have seen a lot of covariations in terms of space, of time, and these covariations can be also linked with a lot of environmental parameters. The temporal effects, we have seen that there is first a pioneer community at the surface of the algae, and then we have new colonizers which can colonize the surface due to the increase of temperature, but also the increase of some algal metabolites like the DMSP. And for the spatial effect, there is a clear effect of the oxidative stress of the copper, and to act against this stress, the holobiont might have an anti-oxidative strategy against this stress. That's all for me. Thank you for your attention. I would like to thank, of course, my PhD advisor, Gérald and Jean-François, and also all the team at Naturalis, where I am working on about sponge microbiomes. Thank you for your attention.
Thank you, Benoit, for your amazing presentation on this brown alga. If there are some questions, please take them in the chat and we ask them to Benoit. And you can also take your camera on to ask a directory to Benoit. So if you have some question, take it on the chat. If we don't have question now, I have a general question. So the temperature is better for the growth of your alga. Why don't study this alga after July, in August, for example, and after in September?
The occurrence period of the Taonia is basically between February and July, and after July, they all die. Actually, basically.
Okay.
So the sporophytes form are all dying, and then during the winter only the gametophyte forms are staying, but in a really juvenile way.
Okay. Thank you. We don't have a question now. I have a second question if we have the time. We can see easy the temporal shift between February and July for the metabolites and other. You found DGTA for the growth of your alga. Do you have some other metabolites more abundant in July and in the other end more abundant in February, for example? In February, do you have other metabolites more abundant in February?
Yeah, so basically most of the VIP that we found linked to the temporality were VIP overproduced during the time, especially in July. There is not a lot of, I think there was almost no VIP overproduced in the first one, at least in February. But yes, there is also other lipids like phospholipids, which were also overproduced over time.
Okay,
but,
thank you.
Algal growth.
Okay, there are one question in the chat of Crystal. Do you integrate all phase of the life cycle of the alga in your studies, and are there difference between gametophyte and sporophyte?
Yeah, so actually for this, so Taonia is very, very particular for the life cycle, because unlike, for example, Dictyota, you cannot really see the sporophyte phase. They are really, really small, and they are basically surviving the winter in a really small form for the gametophytes. And for the sporophyte, you can see it growing and getting bigger and bigger from February to July. So actually, what all the results that I presented was the sporophyte life cycle, the step of the sporophyte.
Thank you, Benoit. If we have don't questions, I can take the lead to Samuel to introduce the next one. And thank you, Benoit, to present and to go in this webinar.
Just before that, Samuel. Okay.
Vas-y.
Okay, just before that, Samuel, take the lead. I will just share my screen 2 minutes because the RFMF is currently organizing the next days, the next meeting days, and it will be in Perpignan, that is in the south of France. So I'm not sure if you can well see my screen. So it's just a pub, some pub. So it will be mainly French speaker, but every presentation will be in English. So if you want to come visit us in Perpignan in France, so you're very welcome. And so you can find every information on the website, and I will write the website on the chat. And that's it.
Just as an additional information, the early bird registration are at the end of next week. So for early bird registration and also for abstract submission for oral communication and flash communication, the deadline is 17th of March, so it's quite close. So don't hesitate to do this quite rapidly to be on time. Meanwhile, actually, the next speaker is trying to connect. We don't know why, and she's not able to connect for the moment to go to meeting. For some reason, we tested in another building, and in the building where she is, it doesn't work yet. So we are trying to find another way. So I will try to go back to her and hope that she will succeed in connecting to GoToMeeting today. Maybe we can wait 2 minutes and then we will take a decision if it's possible for her to do the presentation.
Bonjour Samuel, tu m'entends?
Yes, we can hear
Super.
you.
Ah, thank you. Ah, cool.
Bon, merci.
Okay, Samuel,
On
can you
va
just...
pouvoir reprendre.
I'm very
Donc,
sorry. So I'm on another computer. So maybe you could share my file because I don't have it on this computer.
yes, I can do that. So just to begin, we are very happy to have Dr. Catherine Roullier here, which is actually one of my colleagues from Nantes. So she is associate professor at the Faculty of Pharmacy of Nantes, working on natural products from fungi with a specific focus on halogenated compounds produced by fungi. She's going to present today work on halogenation within marine fungi. Maybe, Christelle, can you give me the right to share my screen?
I gave you them, so you should be able to share your screen.
Oh, yes, I think that's okay.
Yes.
Yes. Thank you.
So thank you, Catherine. The floor is yours. And let me
Thank
know
you.
when you want me to switch slide.
I'm very sorry about that. Okay. So I've tried everything on my computer, nothing was working, so I had to take the computer from the one colleague, so I'm very sorry. So first of all, I want to thank the organizers of this webinar to let me present you some of my work. So in fact, the slides are in French, but I'm going to talk in English. I want to present to you a project that was recently financed by the French Research National Agency about halogenation in fungal, in marine fungi and the access to biocatalysts. So I'm gonna try to show you how Metabolomics helped us in different aspects of the project. Okay, can you switch?
Yes.
So I'm gonna first present to you, okay, I'll try to make it quick. So marine fungi and halogens, then how did we get to this study? What was the workflow and some of the first results, because of course this is still a project ongoing. And finally, I'm gonna conclude on what we obtained. Next. Okay, so first of all, when you talk about halogens, when you talk with your family or whatever, the people around you that are not scientifics, of course, they think about this. Of course, I hope all of you don't think about this, but more about the next slide, which is, okay, halogens are, I think, as you all know, fluorine, chlorine, bromine, iodine, that we can find in different organisms. Next, sorry, Samuel. And of course, you know that those atoms are very electronegative. They lack only one electron on their external part, and from the smallest one, which is fluorine, up to iodine, and this confer to them quite interesting properties. Next. And if you look into natural products, so which are the compounds produced by all types of organisms, in fact, the first halogenated natural product that was described was back in 1896, the 3,5-diodotyrosine. If you look a little bit later, there were two compounds from lichens that were described, chlorinated compounds. And in fact, in 1961, only 29 metabolites from microorganisms had been discovered with halogens in their structure. And up to recently, we have more than 10,000 molecules with halogens in their structure that have been described. So this is a field which is really increasing. We find more and more compounds of this type. And if we go a little bit more into the details, in fact, those compounds, they can be quite interesting for many aspects. And I wanted to cite at least one, like vancomycin, which is a compound which is used as an antibacterial in pharmacy. And this compound, in fact, it has been shown that if you remove the 2-chlorine from the structure, then it completely lacks its activity, its antibacterial activity. So those atoms are quite important for the activity of some compounds. I wouldn't say it's the case for all of them, but at least they confer some interesting physicochemical properties. Next, please. If you look in the different ones that have been described, so in the 10,000s, the first two producers, they are organisms from the marine environment, because we know that in the marine environment there are a lot of halogens, especially chlorine, bromine, iodine. But if you look a little bit after, fungi, which are our subject of study in the lab, they are in the 4th position, so they're quite interesting for this. If we go next slide. So if you think about fungi, of course, you can think about that. But what is important is that fungi, they are filaments or yeasts growing in different ways, either saprophytism, they can be symbiotic, different ways. And in the lab, we're more working on the microorganisms that we can grow on petri dishes. And because you can cultivate them and you can have a look to what they are able to produce. Next, please. And back, I would say, in the 1990s in the lab, our previous director, who's retired now, thought that, and what in the sea, why don't we study fungi from the sea? Because so far there weren't so many studies about it. Next, please. And if you look in the literature, the first species isolated from the marine environment was a Phaeosphaera tifarum in 1849. Then there were some first studies on strictly marine fungi, and the first studies 1944 on wood that was submerged. And I would say the first definition of what is marine fungus was in 1961, and then 1979 was the one that was used by most of the people about facultative or obligate marine fungi. Thank you. Next, please. And if you look in the literature, the term marine fungi actually really increased at the beginning of the years, yeah, around 2000, and it's still increasing. More and more people are working on it, and there are a lot to do in this area, especially in the metabolism. Please, next. And still, people are still. Yeah, okay, it doesn't matter. For just to let you know, at the beginning in the lab, people were working more on the mycotoxins they were able to produce, because we thought they could explain some toxicity from shellfishes, from shellfish. And more recently, we're more interested in what they're able to produce that could provide us some new agents for therapeutics, for example. So that could be a promising for giving new drugs. Next. And one of the interesting compound that was isolated in the lab more than 10 years ago was this compound, which was isolated from one fungus collected in the marine environment near the Loire estuary. And this compound, if you look a little bit deeper, but it's got a chlorine, and it was patented for its very interesting activity on osteosarcoma cell lines. And I would say that was kind of the beginning of the interest in the lab in halogenated compounds. Please, next. So all the studies we're performing in the lab, they are coming from fungus that have been collected. So in this part of France, really on the coastal area. So either from shellfish, sediments, or even seawater, all of those samples have been cultivated. All the fungi have been isolated from this. And at the moment, we have more than 1,000 strains that are available in the lab for study. Please, next. So in fact, regarding the interest of a halogenated compound for therapeutics, in fact, if you look, this is a study from 2014. If you look to drugs that come into clinical assays, into clinical trials, we have around 30% of the molecules that have at least one allergen on their structure. Of course, there are fluorinated compounds that are more coming from synthesis, but still, you still have chlorinated, brominated, iodinated compounds that seem to be quite important in that area. If you look, next please, to the abundance of halogens in the marine environment. Next, please. If you look to the way fungi are able to produce halogenated compounds, this is where our project came from. So are our fungal strains coming from the marine environment, are they interesting to produce halogenated molecules of interest for therapeutics, for example? Okay, next, please. So what we did as a preliminary study was to study different strains from our collection. So not the 1,000 strains, we selected some of them. We extracted, did an analysis by HPLC coupled to mass spectrometry. And we had a look and developed a script to look if those halogens were, especially chlorine and bromine, they have typical isotopic pattern because you have an abundant isotope in nature. For example, chlorine 37 represent around 1 to 3 chlorine 35, and for bromine you have almost a half of the two isotopes, 79 and 81. So this natural abundancy gave us a very specific pattern in mass spectrometry, especially the M+2, which is clearly increased compared to a non-halogenated compound. So this is the example of a spectrum for a monochlorinated compound. And also there is a second aspect, is what we call the mass defect between the M and the M+2, which is around 1.997. So if you do high resolution mass spectrometry, it's also something you can measure. And if you have this type of specific mass spectrum, you can deduce the molecule contain a chlorine or bromine, or even sometimes both. So we developed an automated script that was on R, and that the idea was that from our HPLC-MS data, we could find out which spectra had this specific pattern. Next. And from this study, which was performed on all the strains you can see on the bottom, we could find quite an interesting number of halogenated, I would say more features, but yeah. And all those compounds, we had a look to the databases if they were known or unknown. And we could find, so the unknown are in blue and the known are in orange, and we could find quite a good proportion of unknown halogenated compounds. Next. So in fact, the workflow, so from this from this preliminary study, we thought that was an interesting thing to pursue and have a look to what kind of compounds and halogenated compounds our fungi are able to produce. So this is the first aspect of the project. Next, please. Next, please. Thank you. So the first aspect of the project is to find out those halogenated molecules and especially describe them, characterize them, especially the unknown ones. In the second aspect of the project, we want to look at the enzymes that are able to produce those compounds, because of course in nature most of the organisms use halogenation enzymes to produce halogenated molecules. Okay, next, please. And these halogenation enzymes, they can be of different types. So in nature, there are different ones that have been described. First, the one on the left, those are enzymes that transform ions such as chlorine or bromine into a reactive species. And you have, in this category, you have haloperoxidases coupled to heme, haloperoxidase coupled to vanadium, and flavin-dependent halogenases. So those three are able to make interesting halogenation on electron-rich substrates. And you have also two other mechanisms of halogenation that have been described in nature, either with a radical, with the non-heme iron-dependent halogenases, and nucleophile attack also with S-adenosyl methionine halogenases. Next. And the one that have been described in fungi so far, there are only the three on the left. Next. And in this, from this, VHPO are the one that are, there are only 4 described in the literature, so not much. Next. So for this part, we want to look at the genomes and find out typical sequences corresponding to halogenation enzymes. This is made in collaboration with the LUBEM, which is in Brest, in France, with Laurence Mellecadière, a colleague there. Next. So we hope from this that we're going to obtain different type of enzymes. Next. And also we want to have a look to the regulation and the modulation of the production of those halogenated molecules in fungi. And from this, of course, we're going to generate a collection of extracts. So this first aspect of the project is really fundamental. The objective is really fundamental. So it's to describe and understand the halogenated metabolism in marine fungi. Next, please. From this, of course, if we build a collection of halogenated molecules, we're going to screen them for their biological activity, especially as I said before, they can be quite interesting for therapeutics. The extract collection will also be screened on different assays. I'm not going to talk about that today, but we have developed another script and another strategy that which help us from the biological data that we obtain from extracts or fractions to be able to highlight peaks that could explain or that could be correlated to the activity that we observe. So, but I'm not going to talk about that. So the name is Fibico. And finally, because we have an enzyme collection, we hope to build this. We also can use this in green chemistry to produce new halogenated molecules. And this part has been done in collaboration with the team of Frank Roman in TU in Delft, in the Netherlands. The objective of this second part of the project is more applied. It's to value the results either in health but also in green chemistry. Okay, next please. So for the first results that we have obtained, the first thing we wanted to do is to find out the halogenated molecules. So we already had highlighted some in the preliminary study that I showed you, but we wanted to try also some new softwares, I would say, or new strategies to highlight halogenated molecules to see how complementary they could be. So we used the MiAllocross script that was already used in the lab and developed in the lab, but we also used Enzymine that you probably already know, which is a peak picking tool to get the peaks from HPLC-MS data. And we also tried HaloSeeker, which has been developed in Laberca with Yann Guiton and Ronan Cariou, also to highlight halogenated molecules. And from those halogenated peaks or molecules, the idea is that we can annotate the compounds and find out which known halogenated molecules we have in our extracts. So, but of course, this means having a database of all the, at least all the fungal molecules, halogenated fungal molecules that have been described in the literature. And from this, of course, we can deduce which peaks are halogenated but unknown. In parallel, we have another tool in the lab that is developed by Samuel Bertrand, which is part of the Talasomix platform, and the name of this, I think it's Promanot, it didn't change. And from this tool, we can at least do all the peak picking work up to the annotation, and we have implemented the fact to detect also from mass spectra the halogenated pattern, typical halogenated pattern. And this annotation here is more automated. We also want to look at another way also to detect those halogenated molecules by using HPLC MSMS data. So the idea here is that we can use molecular networks that we can generate with MedGem and GNPS. And I guess you already know that. And this allow us to group together compounds that would have similar MS2 spectrum, and also to interrogate databases to find out non-halogenated molecules. And finally, the last thing that I think about doing, it's not done so far, it's to, please next Samuel. It's to use the annotation that we can obtain from GNPS to find out non-halogenated molecules. Samuel, can you just please? Yes, tell us. Okay, so we can find molecules, non-molecules that are non-halogenated. This is another thing that we can do with GNPS. We can do annotation of non-halogenated molecules, and we could from that also try the new tool that has been developed by colleagues, the colleague Grégory Gentajou, who developed a tool to make reactions on compounds and find out, I mean virtual reactions, and find out if in the molecular network that we have generated, those different compounds could be present. So I'm thinking about using this tool to implement halogenation reactions and be able to find out also from that if we could find in unknown halogenated molecules. So this is the last arrow, please, Samuel. Yes, thank you. So this is the workflow and the different thing we can use to highlight those compounds. So far, we've only, yes, you can. We've only used the three first tools that are more dedicated here to HPLC MS data, so without the fragmentation, the MS data. But still, that was a work that was made by a master student last year. So the two Mialoqua and HaloSeeker, they're quite close, and the idea is that they're really specific into the detection of halogenated compounds. But we also investigated the use of MZMine, which is more a peak-picking tool, but still you can still find out the isotopes and try to find if you have also typical patterns of halogenated compounds. So we compared. In fact, we could see that they're not giving all the same results. And from those different tools, we were able to find out different families of halogenated compounds. Please next. 4 families of molecules have been highlighted from this first study. Yes, compounds from the Griseofulvinster family, and from those compounds, 3 of them seem to be unknown. So another family is the family of Peditrem. this type of quite complex alkaloid, and we could find from our data that we had at least 3 unknown compounds there. Another family is constituted by cyclochlorothene compounds with a very unusual, I would say, chlorination pattern. If you look at the position of the chlorine, with 1 unknown here. And finally, we had 1 strain which was a very high producers of azaphylons. And here I didn't even give you a number because there were quite a lot, and yeah, we didn't. It's not finished yet I would say, to analyze all the data on these azaphylons. So interestingly, from we only investigated 8 strains, and we still have 4 different families and different unknown compounds. So now the work is to go further into the purification and structural elucidation of this compound. On the part of halogenation enzymes, we have highlighted 2 enzymes, 2 VHPO. So as I said before, only 4 from fungi have been reported so far. So we have highlighted 2 more. Next. So we proceeded by genome mining, so either by PCR screening or by looking at the whole sequence when the whole genome had been sequenced. And from the different strains that we had investigated, so some of them had positive PCR screening, and 2 among the 4 that had a positive PCR screening, 2 of them were confirmed to be vanadium haloperoxidases. And next, please. And we managed to produce them in a host, so either Escherichia coli or Pichia pastoris. And to obtain those enzymes, the first one was purified and was active, and the second one was purified but still not active because of folding issues. And this is all I can say about that for the moment. Yes, please. So for modulation and regulation, we are at the moment experiencing a knockout experiment. We want to knock out the genome for 1 VHPO and see how the metabolism is going to change. So please, next. I'm going to go quite fast on this. So the idea is that, yep. The idea is that we want to replace the VHPO gene by a resistance gene, and to compare after that the metabolism of the wild strain and of the knockout strain, and compare the metabolism by, of course, metabolomic study. But this experiment is still ongoing. Please, next, I think. And this is done in collaboration with a lab in Toulouse. Please, next. For the last part, so because we had purified 1 enzyme, we did some assays of biocatalysis, and we did some experiment on very simple substrates. So please, next. First of all, of course, before doing any experiments, you have to make sure that your enzyme is tolerant to different solvents, because not all compounds are soluble in water. And what is very interesting with VHPO is that these are enzymes that you can put with some solvents and they can tolerate them. So here you can see that the best solvents where it keeps most of its activity is ethanol and DMSO, which was quite interesting. If you compare to the other enzymes that I showed you before, they are really more robust than the other ones, and they are more prone to be used for green chemistry or biocatalysis. Next. So we did on very simple substrate like styrene, phenol, fluoroglucinol, we did some experiments. And what we did is that we compared the enzyme we had purified to other enzymes that were available, either by collaborations. And we found out that our enzyme actually act very similarly to the others. It had, interestingly, an optimal pH of reaction at 7 versus 5 for another one, which has been used for biocatalysis. And it had a very high affinity for bromine. Next, please. So if I just come back to this workflow, can you please go up to the end of this slide? Sorry. In fact, no, again, please. No. Yes, perfect, thank you. Samuel, can you please just go at the end of this slide? No. Thank you. Yes. So here, just to come back here, we have this enzyme which is available, and we have found out a way to produce halogenated molecules, but we also have a collection of extracts, and we thought that using this enzyme on extracts could be interesting. Next, please. Because in fact, if you look at the different databases of natural products at the moment, so I just picked up some of them, like Natural Product Atlas or Dictionary of Natural Products, or even Lotus, you have a lot of molecules that have been described. At the moment, we are more than 300,000 compounds that have been described in the literature. Most of the time, many compounds are the major compounds you can find from the organisms that are being studied, and the risk to rediscover again the same molecule begins to become quite high. The more you go, the more is known, of course, and the risk of re-isolating non-compounds increases. And I think this is one of the problems a lot of natural product chemists have, and we are trying to develop strategies to avoid that, and to be able at least to annotate the compounds even before isolating them, just to make sure that if we spend time and money on isolating a compound, we hope at the end it's new, at least that we have new chemistry. So different ways to try to give more chances to isolate new compounds. So one of them is, for example, using co-culture when you're working with microorganisms, because you want to induce new biosynthetic pathways. Another one could be using different culture media, like the OSMAC approach, one strain, many compounds. Another one also is, in fact, treating your extract with a reactant, to change it. And for example, recently, one example of bromination has been performed, and we thought that maybe because we know we have one enzyme that is able to make that, which is not specific to one substrate, because we've shown that it could react with different substrates, then we thought that we could do the same. We could diversify our extract, generate new chemistry by using this enzyme on natural extracts. Next. Yes. So next. So what we did is we used 4 different strains that were available in the lab. We treated them with the enzyme and what the enzymes need. So in fact, hydrogen peroxide and KBr and potassium bromide on the right. We also did the same thing, but without the enzyme. And we had a negative control, which was just the extract without any treatment. And of course, after that, what we did is that we recorded HPLC-MS data and compared the profiles. Next, please. So for one of the strains, so you have here the PCA, the principal component analysis in positive and negative mode for Penicillium expansum extracts. So in red, you have the untreated extract. In blue, you have the extract treated with the reaction medium, but without the enzyme. And in blue, you have the enzyme treated extract in green. for Aspergillus pseudoglockus, the second, the third Trichoderma, and the fourth Ortea wernicki. So as you can see on the first two principal components, this experiment seemed to work quite well for the two first strains because you have the green dots that are quite separated from the others. However, you can see that even the reaction medium had some influence on the metabolites present in the extract, so probably due to the use of hydrogen peroxide, which is a strong oxidant. But for the two others, Trichoderma and Ortea wernicki on your right, you can see that green and blue dots are not so well separated on the first two principal components. So it doesn't mean nothing happened, but something happened at least with the reactants. But maybe if something happened with the enzyme, maybe it concerns not much metabolites. So the reactivity of the metabolites might also be quite important. So just to show you on one of the examples, so please next, the one that worked the most, which is Penicillium expansum. Oh, sorry, yes, I forgot this part. We went, no, that's fine. We also went a little bit further because only the PCA was not informative enough, and we wanted to make sure that something happened did we halogenated something? We did, of course, an analysis with the MiAloqua script that we have developed in the lab. And you can see here that for, at least for the first strain, so MMS42, which is Penicillium expansum, you can see that with the enzymatic treatment, you have a clear increase in the halogenated features that we could detect from the HPLC-MS profile. So that was quite consistent here for the first strain. So we investigated this one a little bit more. Please next. And we had a look to the compounds that were produced. So if you take communisin A, which is the second square, you can see in the middle on the untreated extract that it's present as a peak. It's present when the extract is treated only with the reaction medium on the blue trace. And on the green trace on top, you can see that this peak has completely vanished. And in fact, we could see with the different arrows that we had quite a lot of brominated features appearing on our chromatograms. Next, please. So, of course, one of them was the bromocommunisin A because it's a major compound in the fungus. So we, rather than purifying from the extract because we didn't have high amounts, we actually purified the communisin A, treated it with the enzyme, and we managed to produce this new compound, which is the 12-bromocommunisin A. Interestingly, the conversion rate was 96%, which is quite good. And we confirmed that the one that we saw on the chromatogram was the same as the one that we actually characterized. Okay, next please. So in the end, this strategy was quite interesting because we managed to increase the number of halogenated molecules. And we also compared the number of hits in the databases. So in fact, we decreased the number of hits in the databases, meaning that we actually produced some new chemistry, which was one of the goals. Next, please. So in conclusion, in this project, Metabolomics has helped us to identify in a more automated way from very complex, because when you work with natural extracts, of course, there are complex mixtures. So it helped us to highlight halogenated compounds, new halogenated compounds that are at the moment being isolated to be characterized and to elucidate their structures. And because we have been able to identify 2 new VHPO from fungi, of course, we have had a look to their interest in biocatalysis, and we've been able to produce some new chemistry, even from extracts from complex mixtures. So on both sides, Metabolomics helped us. In the perspective, we're going to pursue, of course, the biocatalysis assays. We're going to isolate and identify all the new molecules that we have highlighted. And in the end, of course, the idea is also to test, do some biological assays on the different molecules that we'll have in hands. Or you can pass that, please. Next, please. So I wanted to thank all the people that have been involved in this project from far on, or all the students that have worked, even in the preliminary studies. And I thank you for your attention. I'm sorry if I've been maybe long. I was quite stressed because of the problem.
Okay, thank you, Catherine, for this very nice presentation of the Allocat project. Does anybody has any questions? Maybe you can write it down in the chat or just ask it directly to open up your micro. Maybe Christelle, you can ask it directly.
I have one question. It's just a curious question. In one of your first graph, you showed that in fungi, the halogenated molecules are have mainly chlorine. But then I saw that for algae, it's more halogenated molecule with bromine. So I'm just wondering if you have any idea why we can observe this.
Yes, it's a good point. That's true. So far, most of the halogenated compounds isolated from fungi, they are chlorinated, but also because a lot of terrestrial fungi have been studied. That's also probably one answer, but maybe also, in fact, we don't know so far. In what we've done, it was the same from our study. We found mostly chlorinated compounds rather than brominated. So yeah, I don't know, but maybe if we, in fact, in the strains that we are looking at, some of them they are coming from hydrothermal vents, so different locations, and maybe from the deep ocean, maybe we'll have maybe more brominated. I've got no idea, in fact. So I think it should be investigated a little bit more.
Thanks.
Does anyone have any more questions? Because maybe there is time for one last question. So I have one.
Samuel, I have one question.
Okay.
Yes, I'm actually wondering if you're at the moment also looking at the same fungi, but from different source, and wherever the compounds you're looking for, they have a function. Because as you know, my first love was fungi of seaweed, so I'm always interested by this topic about the source, what kind of compounds you get there. I know there's many compounds. I had no time to look at all of them, so I'm just wondering if you have more information about that.
Yes, so I didn't say everything, but at the moment there's one of the yeast, the Hortea wernickeyi, the one from which we have isolated one enzyme, which actually we really want to work a little bit more on this one. First of all, nothing has been really done in terms of secondary metabolism in this yeast. And second, we have a collaboration with someone who's specialized in the physiology. She's sequenced all the genomes from 60 strains from the same species. So in fact, we're actually beginning a metabolomic study to see if we have some kind of differences in the metabolism, and if this metabolism is related or not to their genomes. And next also we'll probably look also at the way we can cultivate them in different ways and how they react to that. So this is a project just beginning at the moment, on this specific yeast. Yes.
Yeah, sounds good. Thanks a lot for your talk.
Thank you.
Well, thanks a lot, Catherine. So it's 4 p.m. That's the end of our the webinar session, just right on time. So that's very good. So I really want to thank all the speakers for their very nice presentation, and hopefully we got Catherine for the last one after half an hour of struggling. So that was a very speed moment. So I really want to thank all the person who were attending this webinar for the questions, their presence, and the next webinar will be announced on the RFMF mailing list, and you should get information soon about it. So I think we can stop the webinar now, and
Cette conférence va maintenant être enregistrée.